All right, transcription in eukaryotes is very similar to prokaryotes. There's just a couple of differences in terms of location and how it started and what controls it. But for the most part, RNA polymerase is coming in and functioning like in terms of copying the same way. Now, we're going to talk about the role of RNA polymerase in this. There's actually a couple different RNA polymerases for eukaryotes, and we will talk about the significance of transcription factors, which play a larger role in controlling transcription in eukaryotes. And if you go on to take any molecular biology courses, you will learn all about transcription factors, and I will show you some of my favorite here in a second. So eukaryotes have the nucleus in their cell, right? And the DNA is stored in there. That means we're going to separate the processes of transcription and translation because transcription is going to occur in the nucleus, and then the mRNA is going to need to move outside the nucleus to be translated by the ribosomes. Now, another difference in there is that in eukaryotes, the mRNA is going to be processed. Bits of it are going to get cut out, and some bits are going to get added to it. This does not occur in prokaryotes. So let's start by looking at RNA polymerases. So just as we talked about in DNA replication, there are multiple RNA polymerases in eukaryotes. We have 3 of them. RNA polymerase II is the main one we're going to focus on. That's the one that transcribes protein coding genes. So that's making mRNA. Now, RNA polymerase I, we believe that its primary function is to transcribe rRNA genes. Now remember, the ribosome that's going to translate the mRNA into protein, that is made up of a mix of rRNA and protein. So these genes are transcribed by a special RNA polymerase. RNA polymerase III is also going to transcribe rRNA, but also tRNAs. Those will be important in bringing the amino acids in to decode the mRNA. And also small nuclear RNA genes, which are a type of non-coding RNA that we'll talk a little bit about here in splicing. Okay, eukaryotic promoters are much more complex than prokaryotic promoters. So here's a eukaryotic promoter sequence, and you'll notice that there are all different sorts of regions in here, and also just the transcription machinery is much more complex. There's a bunch of like generic transcription factors that come in and bind to these different regions. But what's really interesting is instead of like the sigma factor, which kind of showed the prokaryotic RNA polymerase where to bind, eukaryotes use specific transcription factors. These specific transcription factors are going to bring in RNA polymerase or prevent RNA polymerase from doing its job of transcribing. So there's generic transcription factors, then there's specific transcription factors. They act like switches, okay? They turn on transcription or they prevent transcription turning it off. We will talk more about this later in a section when we're talking about gene regulation. But why do I talk about this? Why is this so important? Well, transcription factors can control not just one gene, but many genes. And they often get referred to as master regulators. So in the process of development, which is something that I find very fascinating, transcription factors play large roles. So what makes the head or the eyes or the brain or hands and fingers? Transcription factors are behind a lot of this control. And when transcription factors have problems, interesting things can occur. Not always good things, but interesting things. So here are my favorite transcription factor mutants. These examples here are showing you what happens when one transcription factor has a mutation. So I've showed you antennapedia before. Remember, just one protein being non-functional can lead from fly antennae being converted into legs instead of these antennae during the development process. That's one gene, right? Now this shows us that transcription factors control many genes. And there's a transcription factor that controls the patterning of legs. And if you also there's a transcription factor that controls the patterning of antennae. And if you break those, you can get the wrong organ in the wrong place. You can see another one here. Normally, fruit flies produce these little structures called halters. They're important in fruit fly mating. If you break one transcription factor, those halters can get converted into another set of wings. Whether those wings are functional or not depends, but I think you can see that evolutionarily, this could be really important. You could get a whole new set of organs in different positions just by altering the expression of one gene. So there are some insects that have multiple wings. Think dragonflies, right? They have two sets of wings on each side. That could be from just these changes in these master regulatory transcription factors. These genes in fruit flies are called Hox genes. If you take 300 level genetics, hopefully they will talk all about Hox genes. They're some of my favorite things. They're so cool that you can just change one gene and get these whole different structures forming. In plants, we see similar things, and a lot of my PhD work was actually studying these, what we call MADS box gene transcription factors that control floral patterning. So in a normal flower, you have on the outside the green covering called the sepals, then inside of that you have the petals, then the anthers, the male bit, and then the stigmas, the female bits. If you break one gene, called agamus in this case, you get a pattern where it just keeps making petals over and over and over and over again. Now, that's not great if you're a naturally occurring plant because you can't undergo normal plant reproduction. But if you're a plant breeder, that's really cool, right? Because you go to the store to buy flowers, you want lots of petals, right? That's what the market demands, right? You got to have the biggest petals and the most petals to sell your flowers. So these agamus mutants are often what you find in the floral section at your supermarket or something like that. They're mutants that floral breeders have selected for, and they're like, wow, those make a lot of petals. Those look really nice. I'm going to keep propagating that. And luckily with plants, you can propagate asexually, so you can keep these lines going. So these MADS box genes, transcription factors, not only very cool in revealing the processes of development to us, but also in being important if you're a florist. Okay, we're going to look at an animation that shows transcription in eukaryotes here.
The central dogma of molecular biology, DNA makes RNA, makes protein. Here the process begins. Transcription factors assemble at a specific promoter region along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase. It maneuvers the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism. The RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top.
All right, those animations are amazing. They give us like a super unique perspective on this process that we can't actually see. And those are what we actually think this process is looking like. They're modeled off chemical principles and things like that. So transcription, a little bit more complicated in eukaryotes, but basically the same thing is happening, right? So DNA binding proteins that regulate RNA polymerase binding are known as what? Go ahead and pause the video, think about that. All right, hopefully you remember what process we're talking about here. We are talking about transcription, so these are called transcription factors. They bind to the DNA and regulate whether RNA polymerase can do its job or not. So we said there's a few differences in eukaryotes. We saw there are 3 different RNA polymerases used for transcribing the different types of genes. We looked specifically at RNA polymerase II, which is transcribing protein coding genes into mRNA. We have transcription factors. Those are DNA binding proteins that control where RNA polymerase is transcribing genes. And these transcription factors are helping to turn on and off the genes. So they're both positive and negative regulators. We will come back to transcription factors later in the course. Don't worry, they're super cool. All right, that's it for 15.3.